It's my pleasure to introduce our guest speaker for this evening, Dr. Jo Knight. And her path goes from the UK into Canada, and I'm sad to note back to the UK, so I'll bring you up to speed on that. So Jo did her PhD at Queen Mary University in London, 2 fellowships at King's College in London, and then moved to Canada in 2012 to start a statistical genetics group at CAMH, so the Center for Addiction and Health, Mental Health. She has a great group in the city, obviously, 4 graduate students doing a whole variety of projects in applied and methodological statistics, and working particularly with Psychiatric Genomics Consortium. Lots of grants, great papers, etc. And my last phrase, in January this year, she is moving back to the UK to take up a post at the University of Lancaster. So sad. So we're very glad that she is our guest speaker for today. Jo, I turn it over to you. I'm going to get you to mirror your display.
It was that a minute ago. Easier when you can see the slides. So I am going to be looking at my phone not to check my text, but just to check that I'm on time. So I'm not sort of checking email while I'm talking. And also I have quite a significant cold, so I'm probably not going to stay around very long after the talks today, partly because you probably don't want me sneezing and coughing on you, and also because I don't particularly feel like sneezing and coughing on you. But I am based at CAMH, as I've already said, and that's just at the intersection between college and Spadina, so I'm very happy to see anyone and answer any questions another time if that's appropriate, or my email is here and my phone number's on the web, so I'm happy to take questions. I just may not be around for very long today, and actually I've been to this meeting on and off, so I'm around generally. So I'm talking about identifying genetic variants that play a role in complex traits. I'm using the term genetic, I'm using lots of terms very broadly today, and one of the terms that I'm using very broadly is a genetic risk variant, and I will get into a bit more detail of what I mean by very broadly a bit later on. But essentially I'm meaning variations in the DNA, and the variants occur more often in individuals with individuals with a disease than individuals without a disease. And so these variants might predispose someone to a trait like schizophrenia, or a complex trait like that, make them more likely to have that trait, or they might lead to an increase in a measure like blood pressure or height or something like that. So I'm talking about variants in the DNA that sort of predispose individuals to these complex traits. And these have typically been identified by the use, through the use of association studies, which simply look for this frequency difference between cases and control. Now, one of the very common forms of association studies that is carried out frequently these days is the GWAS, or the Genome-Wide Association Study. GWAS definitely stands for Genome-Wide Association Study, and not the Great Western Alpaca Show, which may come up if you search GWAS images on the net. So this is what you should be looking at. So these are the results from a GWAS study. I don't know how many of you are familiar with these commonly called Manhattan plots. Essentially what you have is the y-axis, which gives you some measure of significance, and in this case the higher it is, the more significant it is. The x-axis contains the markers throughout the genome, all lined up. They're often shaded, and the chromosomes are shaded in alternate colours to give you some kind of sense of where you are in the genome. Having said that, as there's 3 billion base pairs, it's kind of squidged, and you can't really tell where you are other than in the chromosome and sometimes in the MHC region or something like that. In this plot here, this is what we would deem a very successful genome-wide association study. There's over 100 hits above the significant threshold, which is a p-value of 5 times 10 to the minus 8. That's commonly accepted, what it has to be to deal with the issue of multiple testing correction. And all those towers in red are the ones that reach that level. And this is the kind of genome-wide association study that people have been using in order to identify these variants that differ between these individuals with and without disorder, or the frequency differs. So that looks great. There's 108 both sides, but there's 35,000 cases and more controls in that study. So that's a lot of time and money. And frankly, the genotyping cost, when you put that much money into recruiting these individuals and phenotyping them, is almost negligible. And everybody knows that the genotyping cost is a lot of money. So it's a lot of money and it's a lot of time. And frankly, that's not enough. So we need to be doing more with these data sets and getting more out of them, and not just accepting, ooh, 100 variants. That, it's just not enough. And what we do know is that this region below that significant threshold, which is so severe because of your multiple testing, is enriched for causal variants. They're not meeting this, they're not meeting their critical criteria, but we know it's enriched. And so what lots of people want to do is try and find ways of sort of going below that line and identifying things based on information other than just a p-value. Because we know, as well as being, although it's enriched for causal variants, there's a lot of stuff at 10 to the minus 7 that's just chance. And so what we and a bunch of other people actually did, all around the same time, is try to incorporate functional information with the association test statistic. So I'm not going to point with a laser because I might blind her, but Sarah, who's on this picture here, is also over there, and she did a lot of the work that I'll discuss in this talk today. So, as I said, 3 people all around the same time published these papers that give us a method that gives us a functional score for each SNP in the genome, which we can combine with the statistical score. And I won't go into how we're going to combine it. What I'm going to talk about today is how we derive that functional score, how that can be different, how the different groups did it, and what we know about the best way to do it, or what we don't know, as the case may be yet. And so this paper here represents a tool called CAD, which people may be familiar with. It's probably one of the best known of the 3 methodologies. This one is a tool called Guava, and we didn't give ours a name, and I think that's why we didn't get such a high impact journal. That was a mistake I won't make again. But anyway, what we all did was put some functional data alongside our association statistics. And so what do I mean by functional data? Well, I mean kind of a lot, actually, because one of the methodologies that I'll talk about had 949 columns representing functional data. So I haven't got tables of that, I'm not going to list them, but I'm going to give you a flavour of what it is that we mean by functional data that we incorporate. Well, lots of it comes from the ENCODE project, which is a project which is basically, I'm going to forget the acronym now, but it's basically an encyclopedia of functional DNA elements. So it's a huge project by many, many labs on many, many cell lines, looking at many, many transcription factors, DNA modification, proteins, and all sorts of things. Because when we look at the DNA, we haven't just got the variants that I've talked about that differ in frequency. We've got all these other things, these other proteins that package and process the DNA. And so this is a figure that is from one of the main ENCODE papers that shows, you know, you've got the chromosome down here, you've got the sort of strings and A's of nucleotides up here, and then you've got histones and then these chromatin and what have you, and then you've got things, transcription factor binding, transcription factors, the general machinery, and all these things bind to the DNA and make the process of the DNA of making the proteins happen faster, slower, being more abundant or whatever. And so this is the kind of functional information we're talking about. We're talking about a variant. Is it a variant that just sits in the middle of the region of the gene that we have, that genome that we have no idea about? Or is it a variant that sits in the middle of a transcription factor binding site, or an enhancer, or something like this, or a EQTL? And we also put in, as did some of the others, some information about conservation, some information about things we already know about genes, like obviously non-synonymous SNPs, because they're the things that we've always been interested in, that change proteins and what have you, but splice sites, all these kind of things. So this is the kind of functional information we're talking about. And this is just a quick picture, because I like it, of how this kind of functional information can be represented on the UCSC browser. But it's all freely available to anyone, and so it can be downloaded in sort of forms that you can actually use, as opposed to just pretty pictures. And so that's the kind of functional information everybody puts together. So, based on the fact 3 groups at the same time did what we did, we wanted to compare them, and so we have this paper that's just come out last month on comparing the different variants. Now, I'm going to briefly tell you what they all did. It has to be said that when we're looking at these 3 that came out within about 6 weeks of each other, there are others, there are quite a lot of others, but we chose to compare these 3 principally because they all produce a score for a SNP to say how functional it might be, how likely it is to be causal. So all of them do that. Other ones, you need to incorporate GWAS summary statistics into the development of the score, so they're not available just to be applied to other GWAS statistics and things like this. So other ones don't actually give you a score for a SNP, they give you some odds ratios for the different annotations. So there's lots of different ways of looking at this, and I'm only looking at 3. There are a lot more, but these are the ones that at the time were giving you these scores for each of the SNPs. So what's the overall procedure? Well, the overall procedure is you give functional labels to pre-classified genetic variants. So if you imagine you have this kind of huge matrix where every row might represent a different genetic variant or a SNP, often indicated by an RS number, if you're familiar with those. You've got all these rows with all these different variants, maybe 2 million, maybe 10 million, depending on which way you sort of cut the cake, but you've got all these rows representing all these different variants. And then you've got a bunch of columns, it could be tens of columns, it could be hundreds of columns, representing all the functional annotations. And you could just have a scoring algorithm that says yes or no, it is in a transcription factor binding site. Or you could have an algorithm that takes into account the read depth, which is the information you get about whether something is in a transcription factor binding site or not. So essentially you've got this huge matrix. In most of the cases, it's the binary matrix that says this is the SNP, yes or no, it does have this functional annotation. But it can, that in itself can vary. And then you've got a classifier that says this is a causal SNP, and this is where it gets a bit tricky, because if I knew what the causal SNPs were in the first place, I wouldn't be doing this, and I'd have a Nobel Prize. But I don't, so nor does anyone else yet, that's why we're trying to identify them. So you've got this classifier that says, yes, this is a causal SNP, or no, it's not a causal SNP. So this is the data, the matrix that you start with, which, as you can already imagine, is going to vary considerably between the groups. Then you derive a functional score from machine learning. So you've got this big matrix, and you've got some known now in there cases of things, SNPs that you consider to be causal and SNPs that you aren't, and all this data, and you're going to use this data to predict the status of SNPs, and therefore unknown SNPs can be given a prediction status. Another confusing point that comes in here that's different from other machine learning is, of course, you don't get any new cases. So if you do machine learning on samples of individuals who have cancer, some form of cancer versus individuals who don't with their gene expression profiles, then you get a bunch of new patients and controls and see if your methodology works. Well, we pretty much know most of the variants in the human genome now, so we don't get new variants, we just get reclassified variants. Currently, there's lots of variants, most of them we don't think are involved in complex traits, a few of them that we think are, and increasingly the number that we think are grows and grows, and the other number shrinks and shrinks, and a couple swap around. But it's a complication that is part of the thing. So you've got this big matrix, you've got all these variants, you've got all this functional information, and then you've got this classifier. And you're deriving from that a functional score from machine learning. So that could be a whole bunch of different algorithms, and indeed 3 groups chose 3 different algorithms. So we'll look briefly at those later. And then you've got to assess the outcome. Now, that seems fairly straightforward, but we did a whole nother, my laptop's gone to sleep. we did a whole nother look at that because different groups were using different ways to assess whether the methodologies worked. And of course, you know, that in itself gave different results, but I won't go into all of that today. But this is your basic plot. But as I've said, you've got all these questions assigned to it. So which variants even should you be using for training? So if you use all the variants in the genome, and then the variants that you classify are a group of, come from a group of variants that have been studied in a certain way, so for example, studied to see in a GWAS context, then the difference between the things that are hit variants in a GWAS context and things that aren't may be confounded by the fact that in the group of things that you're calling not GWAS hits, you've got a whole bunch of other things that were never even investigated as GWAS hits, so you don't even know whether they're GWAS hits or not. So the first question is, should you include all variants? And there's regions of the genome that perhaps you might not want to approach too closely or allow to affect your models too much. So we tend to exclude the MHC region. It has very high recombination rates. There's lots of very high linkage disequilibrium and lots of repeats. And I was told once that studying the MHC region, your reaction to studying the MHC region in detail is like an engineer's reaction when he opens your car and goes, no, that's very complicated. I mean, there's so much going on in there, it's very hard to imagine that it's going to look like the rest of the genome. So you have to think about which variants you're going to build this whole model on. You have to think about what functional data. I've given you a flavour of some of the stuff you might use. But even given the fact you might use encode, as I've explained, there's different ways of doing it. So you might have a binary cutoff, or you might put the quantitative variable in. And which are which in terms of the classifiers? Which variants are causal and which aren't? And how do you even begin to define that? Which algorithm will you use for machine learning? What parameters will you use? And how will you assess the outcome? Okay, so step one, you might be able to read this at the back, but I will go through it, the pertinent details anyway. Step one is you're giving the functional labels to the pre-classified genetic variants. Okay, and so we've got the 3 different papers in the 3 different columns, and then we've got the three different rows here representing the sort of information you need in this step one. So the first thing I mentioned is the non-risk variants. So in our paper, we actually used as non-risk variants all of those that were on Illumina and Affymetrix GWAS panels, because we felt at the time when we were, especially when we were starting this work, although this has changed a bit because imputation has increased our ability to interrogate all SNPs in a GWAS manner, but certainly at the start we felt that, and we're using the GWAS catalogue which contains GWASs that haven't been imputed, we felt that using variants that aren't defined, aren't examined in a GWAS context, and saying that they're not GWAS hits is an oversight because we just don't know yet. In the same way that now, even with imputation, we are examining everything, there's a question of whether we really are examining everything. Because when we try and interrogate a variant that has a very low minor allele frequency, so it's really rare in the same population, if we interrogate that variant in GWAS that has 2,000 cases and 2,000 controls, we're not really interrogating it because we have no power to assess whether that variant has a different frequency in the 2 cases. And so one of the things is, if you use all other variants, you might end up just classifying rare variants if they happen to have a different functional profile than non-rare variants. So in our work, we just use this, the things that were typically on the GWAS arrays, which tend to be more common generally. And then for risk variants, we use the NHGRI, now EBI, GWAS catalogue, and we used the standard GWAS significance level, 5 × 10⁻⁸. This is because these things have been identified in GWAS studies, so we think that's a good basis for thinking they're causal in a complex trait. However, you look at the catalogue closely, and you find that some of the things, the traits that people look in there are things like freckles. And I'm not sure that I think something that's causal in freckles is going to be necessarily relevant in some of the other complex traits. And actually, one of the other things we're forced to do is lump all complex traits together. So I'm also not sure that something relevant in schizophrenia is relevant in cardiac disease, But at the moment, we don't have sufficient data to train very many of these diseases by phenotype. So that's what we did. And then with ENCODE, we used ENCODE data. We only actually used 14 annotation columns. In previous work, we used, I'm going with like 464 as a rough estimate. Sarah will tell you the exact number later. And we tested doing them all separately and clumping them by cell type. And actually clumping them, we got results just as good. And so we used a smaller number. Richie used 174, so he used more than we did, but he'd separated by cell types as we had in our original paper. He used the Human Genome Mutation Database, which is an entirely different way of defining things as risk variants, because they're not from GWAS studies. Most of them have some kind of evidence they're causal, but some of it can be from one sample. And actually there's some now considerable disagreement about whether these things really are definitely causal or not, in the same way that there's limitations with the GWAS. And they used the 1,000 Genome Project, so that's a big project that's assessed lots of variations in lots of different populations. Kirsha used, my computer's telling me to talk quickly. Kirsha also used 1,000 Genome Projects for their null. They simulated their risk variance. I found this very counterintuitive at the time, but I came around to the idea because it worked quite well. They basically said with creative variants, if they're not in the human population, they're probably pathogenic because otherwise they'd be there. So it's a bit more complicated than that. You should go to their paper for the full argument. But essentially, it's a very interesting approach. But it also means their method has an advantage over all the others in that they're able to put a prediction to every single variant that ever might exist, whereas we only do the ones that are currently in 1,000 Genome Project, which is most of them, but not all of them. And certainly not the ones that appear de novo in individuals and cause all sorts of things. And they started with 63 annotations, largely again from ENCODE, but they expanded them creatively to 949 by doing things like splitting up the categorical ones and creating interactions, which is a kind of interesting piece because the other groups didn't use interactions. So you can see how we've all got very different sort of answers to all the questions you might have. We use ElasticNet, which is a kind of logistic regression approach, but it penalises the coefficients because you might be using lots of different variables, and often with logistic regression, if you use too many variables and they're correlated, it kind of falls apart. So this is a penalised approach that handles that. Richie used random forest, which is lots of different decision trees. And Kirsha used support vector machines, which you create a hyperplane in multi-dimensional space. If you have a linear kernel, it's kind of quite straightforward. But you can get much more complex kernels. When we did our comparison, we only used a linear kernel, but you can that can be adapted, so something that could be done further. Okay, so in terms of assessing the outcome, like I mentioned, we did do a quite big investigation into this, so you can look into that further if you're interested, and I've just cited it there. But we came down to the conclusion that mainly violin plots and receiver operating curves, which I'll show you some more of later, are the best approaches. Violin plots are just visual, and they show you, they're kind of a histogram with also the kind of, you've got mean displayed in them and some interquartile ranges and what have you. So they give you quite a lot of information, but they are only visual. Rocks give you a statistic for how much better your model is for predicting whether something's a causal variant or not than chance. We tried some statistics like the Mann-Whitney U, but actually we found that everything was significant, and it wasn't even the ones that clearly weren't separating at all well came up as highly significant. So we moved away from these and took these approaches to our statistics and our visualization. But then there's the next question of, okay, so that's your methodologies, but how are you going to do it? Well, the standard machine learning approach, of course, is that you have test data. So you have your training data, which often is about 60%, and then you have your test data, which is often about 40%. So you make your model in your training data, you test your model in your test data, and you use you get all of your area under the curves, which is the stat you get from an ROC, and your violin plots. You do that all in test data, so you're not cheating. You're doing it on an independent data set. But a test data set isn't quite what we're going to be seeing in real life. So in addition to the test data set, we also worked with what we call kind of more real data set. I mean, it's all real, but this is more what we would see in an ordinary environment. And we worked with the large schizophrenia GWAS data. So there were 2 rounds of the GWAS study. The second round, like I said, had about 35,000 cases and 100 odd controls, and the first round had, you know, something like 20,000 cases or something a bit smaller. And so what we did is we took the, we had a look at the variants that we know were GWAS hits in the second round, in the larger data that had more power, and we determined what our models would do with them, or how our models work with them on the smaller data set. So in the smaller data set, they didn't show up as significant, but we tried to see whether the p-values generally were more significant than they were by chance. So we kind of wanted to see if we could have done what they did with a larger sample just with our methodology. Unfortunately, we couldn't quite do what they did with a larger sample, but we can see that all of these methodologies actually, to a certain extent, do enable you to increase your power without increasing your sample size. And so, obviously, when we did that, we didn't have the schizophrenia GWAS hits in the training data. We took them out, because otherwise that would really be cheating. So, some results. What kind of functional data should we be using out of the different 3 from the test data? Well, it was Kircher elastic net, but it's not striking. Okay, so for this work, we had to hold the classifier constant, otherwise you can't compare. So in all of these, we're classifying from the GWAS catalogue. And then in the columns, we've got the annotations from Galliano, Ritchie, and Kircher. In the rows, we've got the annotations, the machine learning approach to elastic net. Here we've got random forest. Here we've got support vector machines. As you can see, there's not one single method that can definitively separate causal variance from non-causal variance, which isn't sort of a great surprise, because this is just the beginning of people trying to apply this functional data to it. The AUCs range between 0.64, so the area under the curve, 0.64 and 0.71, I think. So low to sort of mildly exciting. Here, I said Kircher with support vector machines and elastic nets were the best ones, and you can see on these violin plots that the means are quite well separated, but the distributions are hugely overlapping. And the one that did pretty badly down here, the means are hardly separated at all, and it's very, very skewed. And so in our test data, we find that, yeah, there's a winner, but it's not, you know, they didn't win the race by that many miles. And actually, the winner's not any of the original contenders, because it's the Kircher annotations, but with the elastic net procedure. So it's a mixture of 2 of the original approaches. And then what about in the functional data? Well, again, we find the Kircher annotation set's doing quite well, and that time with elastic net, but again, it wasn't a real home run winner. But when we looked at the data from the real data, and actually this time we used the predictive scores that were actually in the original paper. So rather than deriving it based, this isn't all equal this time, so we're not using the same classifier or anything. We're using what they've put on the net, essentially. So all of these groups have put prediction scores on the net. So using what they've got on the net, what happens in the real data. Now, I fully disclose I picked this plot carefully because it shows ours doing really well. And I fully disclose that ours is not the best method. None of the methods really come out as a massive winner, but it's, you know, I have that privilege because I'm here. As long as I admit it, I understand we've got doing nicely. And it's a good demonstration of what we do. So this is a QQ plot. If you can see it, the grey in the middle is where all SNPs lie. These are the SNPs on the x-axis here. That's their p-value from the first GWAS study, the smaller GWAS study. And then in the other side, we've got the p-value from the biggest GWAS study. So if the line goes above the grey bar, it means that that set of variants have higher p-values than you would expect by chance. If it's close to or lower than the grey bar, then that set of variants is it doesn't show anything very interesting. And the blue line up there with the circles are the ones that we defined, the top quartile of the ones that we defined as most likely to be GWAS hits. And so, in fact, ours appears to be showing a greater enrichment of a greater ability to identify things that will later demonstrate to be causal, and so that was kind of nice. But like I said, I picked one plot because, as you can imagine from how I'm describing, there are many, many ways of comparing this data, and we had to do kind of everything, every different possible way. So I just wanted to show you like a different way of looking at it as well. So, so we've got the functional data, we've got that, the test data, we've got Elastic, Net and Kirsch winning, but it's not striking. Again, we've got Kirsch and Elastic doing quite well with the real data, but no real winner. What about the classifier? Well, interestingly, when we work with the HDMD classifier, so this time all of the annotation sets and all of the machine learning algorithms, so kind of a 9-set combination, but this time classifying with HDMD, we find that the AUCs go up in the test data. So there is some kind of increased ability to identify the variance, but it's not completely clear that this is definitely a good thing, because again, we're working with test data from the same set. So although this works within its on its own test data, it's not clear to us it works well in the GWAS context. The reason we tried initially to go to HDMD is partly because it had been done by the other group. They also use a database called ClinVar, but partly because we don't want this work just to work for GWAS. We want it to work for next generation sequencing projects. And we felt that the HDMD database might look a bit more like what we would find in a sequencing project, because in fact we can't find any large sequencing project with big enough successful groups of hits for us to do training on. So we tried that. But so there's some kind of thing going on there, but again, it's not clearly doing much better. It's not clearly a good result. And the which algorithm is definitely no clear winner there. This is a very busy correlation table, but essentially what I'm going to tell you is that here, if you use, in the diagonal kind of groups of boxes, you've got the annotations with all the different machine learning algorithms, and the correlation within groups there is a lot higher than when you start going across. And so essentially it almost seems that the annotation is, when you use a different machine learning algorithm, it doesn't make that much difference to what your results are. It makes a lot more difference when you use a different annotation set, which kind of makes sense, other than the fact that the annotation sets were hugely overlapping, so we all work from ENCODE. But it's an interesting result nevertheless. And so one final result, one question I had that up there on was which parameters. So when we did our comparison, we largely tried to compare what the original authors had done and see if we were getting similar results to them. But one of the things that we found was that in the paper that used the random forests, they had done a couple of things which were not standard for random forest analysis. And so one of them was that the final node, you get those decision trees and they go down. And usually you pick a node size, you say my node can't get smaller than whatever, because otherwise your data overfits. It just kind of says, I'm going to put this person in this group because they're wearing a green shirt with lace on, and that obviously means all women wear green shirts with lace on, so it kind of has this like really overfitting problem. And actually that group had done that, so although the method, we're not saying that it doesn't work, because that would be bad, it didn't, in our hands we saw a lot of overfitting certainly in the training data, and then the fitting the test data was not as good as it was otherwise. And so we changed the parameters on that one. And so even when you look at these algorithms, and like I said, with the SVM, we only use the linear kernel, but there are other kernels as well. So we, by no means, have explored all the different options here, and we explored some of them, but you can't do everything. So my conclusion is there's some hope, but there's a lot more to do. And so this is a space that a lot of people are working in at the moment. Lots of people are very interested in this concept, but we are getting bombarded with new data. So the ENCODE data is just kind of the best thing since sliced bread in many, many ways. It's an unimaginably large data set that is freely available to everyone, and it's got standardized procedures, and you can talk to people who generate it. Like there's issues that we found that we couldn't solve, like strange integer preference when you look at the raw files and things like that. but it's just an incredible resource that everyone can use. But there are many buts with it. It's on cell lines which have been immortalized. So who's to say that the transcription factors are binding where they should be? Because these cell lines are not normal anymore. And it's not at different developmental stages. Also, the variants that we look at a variant because it happens to be within a transcription factor binding site without determining whether or not it's affecting the transcription factor binding, because we just don't at the moment have the capacity to do that. So I think this is kind of hopeful. All these methods at the moment make a difference, but they're working with quite muddy data. But that's changing. So there's roadmap, there's psych encode, which gets brain tissues. There's UK-BEC, which gets EQTLs from different parts, different regions of the brain. There's new methodologies that actually not only define that a SNP is within a transcription factor binding site, but there's allele-specific differences in the binding of the transcription. And all of these things need to be incorporated, hopefully, when they do, they will improve these models and they'll really show a much more incremental improvement in identifying causal SNPs. So, just briefly, I'm going to embarrass all my other students who I also went blind for laser. Kartikay is working on a very different approach to this, but wanting to do the same thing. We want to identify causal variants. But he's looking, and this is a little web tool he's created, which we can't quite get public yet, but we're working on it. He's creating a tool where we actually try and identify whether there are motifs that occur more frequently in and around the site of causal variants. Now we're doing this because GWAS is hypothesis free, it looks over the whole genome. ENCODE isn't as yet because we can't investigate things that bind to the DNA if we don't know that they bind to the DNA, and we can't investigate motifs that we don't know yet are motifs. So essentially this is looking for new motifs. This is looking for patterns in the DNA that nobody knows are functional yet. And so that's what his work is doing. And Sejal is doing, who's not here so she won't be embarrassed, but she's doing a project where we try and incorporate information from gene ontology in order to basically work with GWAS data sets and try and improve our power to get findings. So that's just a different kind of functional information that we might bring in. And these are a couple of pictures of people in my group. So thanks to all of them for doing all of the work that I get to talk about. Thank you.
Questions for Joe?
Hey, very nice talk. So, since there was no clear winner on the algorithm, and you have them all working now yourself, have you thought about doing some sort of ensemble method to combine the three algorithms?
Yeah, we've thought about it. We haven't gotten further in that. It's come up as a possibility, but we haven't done that. The other thing that we've thought a bit more about combining, but still don't know how to do, is combining the classifiers. So we've got these different types of classifiers, and we do it on either the HDMB or the GWAS catalog or ClinVar at the moment, but perhaps we should combine those. So we haven't, but it would be a good thing to do. So combination. I think I'm more focused on getting new data in, because the fact that none of the algorithms kind of outcompete the other at the moment makes me think and none of them are brilliant, makes me think that the data's not quite there yet. But
Did you evaluate which one is easiest to use or most accessible? Or?
Oh, obviously ours. No, I'm Sarah will be the best person to answer that question. Sarah, which one did you find easiest to use, other than ours?
Yeah, well, I found that Wahoo is actually the easiest to use because, for example, you have to give a VCF file, and they have a certain version that they are missing like a whole bunch of SNPs, and they can have SNPs appearing multiple times if it appears in like more than one gene transcript, which I just found confusing. With Wahoo, you just give it the RSID. Any SNP can be found in genome, but it's probably really useful, really clear, and easy to use.
What if you get worse results?
I guess linked it with the fact that that one does it, it can do it by position. The one that you said is more complicated can do it by position, so you can't use an RS number because it can do anywhere. So there's got to be a more complicated way of getting anywhere in the genome. Did you want to answer that? Was it?
I was just say no, it's not the word. It's just that the one that we found it, their model is overfitted. So we're about right considering the overfitted.
Have you had any responses from the other two groups to your comparison?
No, we actually wrote them all because we decided that was the best approach. So we sent them all, but we put the paper on bioarchives and we wrote them all and said, this is what we want to publish. We, as Sarah, had interaction with them to get the things working so that they knew they were working on. Someone came to a poster at ASHG, didn't they? But we haven't had much response, which is kind of interesting, particularly because one of them did kind of raise an issue. But we raised it very gently because I think it's not a huge issue, it's something interesting to talk about, but it didn't have a huge effect on the results at this stage. But so they know about it for sure. We also wanted to make sure that they felt we represented them appropriately, so I wanted to give them a chance to see it first, because in case we had some major misunderstanding of their methodology, I don't want to put paper out there that another person's then going to say that you did, or you did, or So yeah, so they know about it, but we haven't had much response from them.
If there's no further questions, please join me in thanking Richard and Joe for their first TorBUG of the season. Per usual, there is the social in the next room, in the black room, so please join us to restart the season.